Hello students, in the last video we have completed the process of electrophoresis. Hope you have understood. In the process of electrophoresis, what we have collected, the gene fragments, the desired gene fragments we have collected. Now the next step will be the making the host recombinant. That means we have to insert the gene fragment into the vector, and after that we make that rDNA or the recombinant DNA, and then the recombinant DNA will be inserted inside the host for its multiplication so that we can acquire the desired product. So this is the sequence of the biotechnology. How can we extract our desired product? But here what is happening, that whenever we completed the process of electrophoresis, what happened, that we have collected the gene fragment. So those gene fragment, if we allow them to enter inside the vector, and a number of time this whole process can be failed. So with that whole long very complicated steps, as we have completed the gene fragments, at least we need to have some copies so that we can able to insert inside the vector successfully, because lots of the attempts can be wrong or can go wrong. So that's why for the gene fragment which is to be inserted inside the vector, we need to have the copies. Then only we can able to insert our gene fragment inside the vector. Then we will make the rDNA, and then we will allow them into the host. So what happened for the multiplication or for the hybridization? We have one step, we have actually one process, one complete technique, that is the PCR. PCR stands for polymerase chain reaction. So what is the meaning of polymerase chain reaction? That here we can have just a gene fragment or a copy of DNA. Then after that, if you allow it to enter inside the PCR machine, then it will give you the multiple millions of copies. Suppose this whole process is going on for about half an hour, then it will produce thousands, millions of copies of the same DNA. Then after that, we can successfully apply that gene fragment. We can allow them to enter inside the vector. If 2-3 attempts are failed, then also it's okay. We don't have to worry about that the gene fragment. If that reaction goes wrong, then the whole process once again we have to do it, right? So that's why we have this whole technique, that is the polymerase chain reaction. So what is the polymerase chain reaction? It is the process of hybridizing our desired DNA fragment or the gene fragment. So we can take one gene fragment, and after that we can get the multiple copies, just like the Xerox. We can Xerox 1-2 pages, suppose, then how many copies you want you will get, isn't that? So same thing for the DNA multiplication, for the DNA copies of DNA, we can go for PCR machine, PCR technique. So this PCR, the whole technology actually have the revolutionary advantages, that it is useful in lots of researches like mutation study we can able to do. Then for lots of genetic engineering processes also PCR is used. To detect a lot of diseases in our body, we can able to go for PCR. For the future detection of the diseases, we can go for PCR. Lots of advantages are there of PCR. These are the application in genetic engineering, in DNA fingerprinting, in research. All this, suppose for the detection of the diseases, lots of viral or bacterial diseases, we can able to go for PCR technology. So this way we can able to do multiple copies. We can form multiple copies from this technique, that is PCR. right? So this PCR that was first developed by the scientist is Gary Mullis. So remember that scientist name. This is Gary Mullis, who developed the concept, the whole technique, that is PCR. Now, how does PCR look like, or how are for the process of PCR, what are the things they are needed, right? So first of all, what we are going to need it, we will need the gene fragment. Okay, suppose we want to make the copy of this, obviously we will need the gene fragment, isn't that? So for this gene fragment, what we are going to have, we are going to have the gene fragment first of all. So this gene fragment will be needed, and for this PCR, suppose these are the requirements. Okay, so let us see, these are the requirements. So what are the requirements? First of all, we need to have the gene fragment or the DNA segment of which we want to make copies, right? So the requirements first of all is the gene fragment or the DNA fragment. So the DNA fragment, what we can call it, this DNA fragment will be known as the template DNA. Okay, remember that. That means here of this DNA fragment, of this template, we want to make copies. So first of all, in this requirement, what we need to have the gene fragment or that template DNA. After that, what we need to have numbers of, suppose this is one enzyme, okay, and that enzyme, this is called as the DNA polymerase, right? So DNA polymerase is one very important enzyme that is needed for the PCR procedure, because you see in this PCR procedure what happened, we actually will work this in the high temperature, like 90 to 90 degrees Celsius it will be needed. In that high temperature, our enzymes, that will not gonna work. So that's why we require some enzymes which will work also in very high temperature, that is around 90 degrees Celsius. So how is it possible? Actually, enzyme, that is DNA polymerase, which is also known as stack polymerase. Okay, so this stack polymerase, that is actually extracted polymerase. Okay, so this stack polymerase is extracted from one thermophilus bacteria. That means that bacteria can survive also in very high temperature, like 90-95 degrees Celsius they survive. So their DNA polymerase also work. If the whole organism, whole bacteria can survive in very high temperature, obviously their metabolism is okay. That means their genetic material, their enzymes, all of them, they are working properly. So this DNA polymerase or this stack polymerase, it is extracted from one thermophilus bacteria. So what is the name of this thermophilus bacteria? That is Thermophilus aquaticus. Okay, so we require one thermophilus bacteria, that is Thermophilus aquaticus, that is a DNA polymerase we need to have for the procedure of PCR. Then the third, what we need the nucleotide, isn't that? Okay, we need to have the nucleotide, small nucleotides we need to have. Then only it is possible that we can able to make the copies of DNA. So here, this is the, suppose lots of small nucleotides are here. Okay, these are the nucleotides. You know what is nucleotides, right? Nucleotides means it is going to have one pentose sugar. Along with that, it is going to have this phosphate, and here this one have to have this nitrogenous base, right? So it is going to have one sugar. That sugar is the pentose sugar. Then it is needed to have the phosphate and the nitrogenous base. Then it will be a nucleotide. Next one, the fourth step. Okay, the fourth requirement. The fourth requirement is the primer, right? So what is the primer? This primer is actually around a very small nucleotide, but it is the nucleotide which is complementary to the gene fragment. Suppose if you see this template DNA. So in this template DNA, if you see, this is suppose A, T, G, C. These are the nitrogenous bases are present in our template DNA, right? So that primer that should be present, that should be needed for the PCR will be complementary to the template DNA. That means here what we need to have, for A we need to have T, for T we need to have A, for G we need to have C, and for C we need to have G. So this one, that is actually one complementary strand of the template DNA, isn't that? So that will be our primer. So what is the primer? That is actually 18 to 20 nucleotide that is complementary, to the template. Okay, so that whenever that DNA fragment will get separated, our primer can attach to the template DNA so that we can have the copies. So simply the procedure is like this. So first of all, this is the template DNA. So the template DNA will be removed by breaking the hydrogen bond. Then after that, the primers, they're going to be added. So here these are the primers. So once the primers are getting attached, then this DNA polymerase, suppose this is the DNA polymerase, here this is the DNA polymerase, this will help in addition of the nucleotide bases. It will help in the addition of the nucleotide bases with the template DNA so that we can get 2 copies of gene or the 2 copies of DNA fragment. So this is very simple and easy procedure technique, that is PCR. Now we will see in detail that in this PCR we are going to have complete 3 separate steps. In these steps, different temperatures are maintained so that our process of making the copies of DNA can be possible. So let us start that one. Now after that, let us see how many steps are there in the technique PCR. So what are the steps? See, in this PCR we have 3 steps. The first step, this is denaturation. Second step, this is denaturation step. Second step is the annealing, and the third step, that is extension. Okay, so in this PCR technology we have 3 basic steps where the DNA will be amplified, the DNA will be multiplied. So first of all, just see what is the denaturation process. So first of all, what we have, the DNA fragment. Yes, so DNA fragment 1.1 end, that will be 5 prime, another will be 3 prime. Here this is 3 prime and 5 prime. So in this denaturation step, what happened, whenever you subject this DNA fragment in high temperature, so suppose the high temperature is 94 degrees Celsius, okay, around 90 to 94 degrees Celsius, 92 degrees Celsius, what happened, in between the nitrogenous bases, what is present? We have the hydrogen bond. So this hydrogen bond will be broken in such a high temperature, that is 94 degrees Celsius. So whenever the temperature is very high, in this denaturation process, the hydrogen bond breaks. Okay, and whenever this hydrogen bond breaks, what happened? These DNA fragments will get separated, right? So here we will have one strand here and the another strand here. Okay, so what we are going to have, 2 strands of the DNA, and each strand, what we can call it, this will be known as the template DNA. Now from these templates we can able to get the multiple copies. Now just see the next step, that is the annealing step or hybridization step. So in this annealing or hybridization step, what happens? See, already what we have this 5 prime, 3 prime end, and here this is 3 prime, 5 prime end. So here the nitrogenous bases are there, right? So here also the nitrogenous bases are there, okay? Now you can see that when the DNA fragments in which end a new DNA fragment will be attached. See, in this 5 prime end what we have? Phosphate group. So with this phosphate group, a new template, a new primer, that will be not added. It's not possible. But towards the 3 prime end, what we have? A free hydroxyl group, right? So if you know the structure of DNA, then only you will be able to understand properly. But otherwise, if you remember this way also, it will be okay. So in the 3 prime end, what we have the free hydroxyl group. So with this free hydroxyl group, the new templates that is of 10 to 18 nitrogenous nucleotide bases, they will get attached, okay? So this will attach here, okay? So this is complementary, right? So if it is complementary, they will get attached to it. So what is this? This is the primer, okay? Remember that this primer will be added here. So the next one, next step, that is the extension process. So in this extension process, what happens? In this extension process, what happened? Already you have the DNA fragments, okay, the templates. These templates are present, right? So these templates are already attached with whom? With the primers, right? So here a primer is added, another primer is added here. So this primer on this end, this will be 5 prime, this will be 3 prime, here it will be 5 prime, and here it will be 3 prime, right? So already the primers are added with the templates DNA. Oh, sorry. So here it is on this side, and here it is on this side, okay? So already it is there. In this extension process, or in this extension step, what happened? That the that enzyme, that is the Taq polymerase, this Taq polymerase will help in the addition of the new nucleotide with this primer. So with this primer, the new nucleotides, they will get attached to it, and it will form a DNA strand, okay? So this way the process of extension occurs. If it is the 5 prime, this is the 5 prime and 3 prime end, right? So in this, what happened? This is 5 prime, in this direction this is 3 prime. Here in this case, this is the template DNA of 3 prime and 5 prime end, and the new nucleotide will join, add to this template from which direction? From this direction, okay? So in this, the direction will be from left hand side to the right hand side, but here in this above template, it will be from this left hand side to the right hand side. So that's why the DNA segments, you know that now, they are called as anti-parallel, because they are not projecting to the same, because they are in opposite direction, they are going to get attached to it. So this is the process of extension. So we get the 2 DNA strands. One is this DNA strand, this is the first copy, this is the second copy, this is the first and this is the second copies. So how many copies we get? 2 copies we get. Within seconds this will happen. Now let us see that in which temperature this process occurs, okay? So just see, if it is the process of annealing, this annealing process or hybridization process, that will occur in what temperature? The temperature is around 50 to 65 5 degrees Celsius. In this temperature, what happened? Annealing process occurs. So temperature variation you will see in different book. You will follow only the NCERT. Whatever the temperature is given in NCERT, that same temperature you are going to write. So in this annealing process, the temperature will be obviously below than the process of step of denaturation. And this extension process, when it occurs at the temperature of 72 degrees Celsius, denaturation occurred in 94 degrees Celsius. So these temperatures that will be needed very much important. Okay, so this way extension occurs. And in this extension process, some another type of some elements, some enzymes will be also needed, like magnesium ion is needed. Okay, magnesium ion is needed. Then deoxyribonucleoside triphosphate, this type of enzyme. Then this is the tag polymerase or the DNA polymerase that will be also needed for this process of extension. So magnesium ion, deoxyribonucleoside triphosphate, then the third tag polymerase, it will be needed for the process of extension. So just see, within seconds what we get 2 copies from one DNA segment. We get the 2 copies, then after that this process, this is just like a cycle. So with a cycle this will go on, and after that we can get millions of copies. How many copies you want, we will get it, because after this extension process, once again the temperature will rise to around 94 degrees Celsius. So these 2 DNA fragments that will break, the denaturation will occur. Once again this 4 templates we will get. With these 4 templates, once again annealing hybridization occurs, and in the extension we will get 4 copies, then 4 to the 8 copies, 8 to 16, 16 to 32, 64. This way the DNA will be multiplied or DNA will be hybridized. So this is the process technique of PCR. Remember that. What are the application of PCR? So application, see, it is very much important, but yes, remember that this is important. What are the application? But when we understand the application, then only we will have interest on PCR, isn't that? If you don't know the use, what is the use of that understanding of this PCR technique, isn't that? So what are the applications? See, in this DNA fingerprinting, okay, DNA fingerprinting, we need the process that is PCR. That means suppose in the crime scene just a hair is got, okay, as a sample. So how can we able to detect the criminal from this just the hair strands? We have to multiply the copies, isn't that? We have to multiply the gene fragment, then it will be very easy to find or to detect the criminal out of this the whole crime scene. Then in the future, detection of diseases, for that also PCR is needed. Then after that, detection of some pathogenic diseases like bacteria or the viruses. Okay, because suppose it is also possible that the bacteria they are found inside our body is very less. So that's why in the normal blood test or the normal plasma or the serum test, it is not able to found the correct or the bacteria we didn't get. So we have to culture it, or we can go for PCR simply, so that we can able to easily identify what bacteria that individual has. Then only we will have the earlier treatment. Then after that we will have in lots of other processes like in genetic engineering, okay, for the study of mutation. Okay, for the study of mutation. Mutation is a very good topic to study in future in the genetic engineering. Mutation carries lots of information that will be very much important to understand. Next one, the other like in researches, Okay, so lots of other researches, this technique PCR will be used. So that is the process of PCR. I hope you understand. See, in this whole PCR, the hybridization or annealing or the multiple copies we get, we will just amplify the DNA segments into multiple copies. So for that, we will have the 3 steps: denaturation, annealing and extension. Denaturation occurs in around 94 degrees Celsius. Annealing will occur in 50 to 65 degrees Celsius, then extension will occur in 72 degrees Celsius. So this way we will have this technique PCR. I hope you understand. Thank you.